Shri Sai Ram. Dear students, welcome back to the 10th class State Syllabus Science Textbook 2 and the chapter Heredity and Evolution. And today's part 4, which mainly deals with DNA, deoxyribonucleic acid. Before we pass on to discuss about the part 4 of this chapter, let us recap the previous part key points and also check out the assignment 3 questions answers and get it cleared. In our previous class, we understood Gregor Johann Mendel, his contributions in the form of 2 experiments which he conducted, monohybrid cross and dihybrid cross, where he selected the garden pea for his experimentation. In the monohybrid cross, a cross between 2 plants which differed in one character, and a dihybrid cross, a cross between 2 plants but differed in 2 characters. And finally, his experimentation brought the result that a monohybrid cross, the phenotype ratio is 3:1, and a dihybrid cross, the phenotype ratio is 9:3:3:1. Further, he proposed the laws on the basis of his experiments. The first, law of segregation; the next, the law of independent assortment; and the law of dominancy. These are the 3 laws that were being proposed by Gregor Johann Mendel on the basis of his experiments on garden pea. Answers for assignment 3. The first question: how do Mendel's experiments show that traits are inherited independently? Mendel's dihybrid cross experiment shows that traits are inherited independently. The example, if we take, in F1 generation, all were dominant characters is being expressed. The gametes of this F1, which were led to self-pollination, we got 4 varieties of pea plants were obtained. In all these 4 pea plants, the heredity characters were present, which shows that the traits are inherited independently. I hope it's clear for all of you. The second question: prove that the dihybrid ratio is 9:3:3:1 with the help of Mendel's experiment. Beginning, you shall write the dihybrid cross definition: a cross between 2 plants which differ by 2 characters. To prove the ratio, the dihybrid cross selected character, stem length and position of flower. Parent plants, tall and axial, because 2 characters should be present. So, tall plant with the flower in the axial position. Dwarf and terminal, the flower is in the terminal position. So, 2 dominant characters and 2 recessive characters. The gonads, capital T, capital T, capital A, capital A, small t, small t, small a, small a. The gametes, the diploid number reduced to haploid during meiosis. So, the capital T, capital A, small a. They undergo cross pollination, fuse, and the zygote is being formed, the F1 generation. We got all the plants were tall and axial flowered. The F1 generation undergone self-pollination, and the self-pollination, the gametes of F1 generation, capital T, capital A, capital T, small a, small t, capital A, small t, small a. And this is the checkerboard which we have written. So, checkerboard. Just writing all the 4 in the box, multiplying capital T, capital A with all these 4 varieties of the gametes that are obtained. Capital T, capital T, capital A, small a, capital T, small t, capital A, capital A, capital T, small t, capital A, small a, capital T, capital T, capital A, small a, capital T, capital T, small a, small a, capital T, small t, capital A, small a, capital T, small t, small a, small a, capital T, small t, capital A, capital A, capital T, small t, capital A, small a, small t, small t, capital A, capital A, small t, small t, capital A, small a, capital T, small t, capital A and small a, capital T, small t, small a, small a, small t, small t, capital A, small a, small t, small t, small a, small a. So, these are the variety of the seeds that were being produced. We took only 2 characters and we got variety of the plants which you are observing here. The 4 varieties we can observe among all these seeds when they were sown. Where among the 2 T's, if 1 T is present, then it is tall. Among 2 A's, if 1 A is capital, then it is axial. Then the same among 2 T's, if 1 T is capital T, then it is tall. Among 2 A's, we have only small a, then the terminal positioned flower. Among 2 T's, if the T is small t, then it is dwarf. Among 2 A's, if the A is capital A, 1 at least, it is dwarf and axial. Among 2 T's, if the T's are small, then it is dwarf. 2 A's are also small, it is terminal. So, the ratio 9:3:3:1, where we had 9 of such varieties and 3 of such varieties and 3 of such varieties and only 1 of this, where you can observe. I hope you will have done this work perfectly. If not, never get depressed. Practice by selecting the characters repeatedly. Surely each one of you will do perfect. It is very simple mathematics. The next part, that is the part 4 of heredity and evolution, is about the DNA, and the genes also continues along with that. We all know that the cell is the basic structural and functional unit of life. The main part of the cell is the nucleus, which is like the heart of a cell. This nucleus controls and coordinates all the activities of a cell. It is the nucleus which is going to make the cell to function and to live in a healthy way. This nucleus contains the nucleic acids and also the proteins. The nucleic acids such as DNA, which plays a major role during cell division by itself undergoing replication, that is division of DNA. Why the cell undergoes division? To increase the number of cells. When do the cell undergo division? When the cell reaches the maturity. So, the DNA during cell division undergoes replication. The RNA is the another nucleic acid that is found in the nucleus, helps in protein synthesis. Then what the abbreviation? DNA, deoxyribonucleic acid. RNA, ribonucleic acid. DNA, deoxyribonucleic acid. RNA, ribonucleic acid. DNA in detail to the IQ level and to the 10th class extent we shall understand, because the DNA information changes and increases as we go to higher classes. DNA, it is a macromolecule which is made up of large number of nucleotide units. It is made up of large number of nucleotide units, and hence it is termed polynucleotide. The DNA is a polynucleotide. It is a macromolecule. Its abbreviation is deoxyribonucleic acid. It is one of the nucleic acids that is being found in the nucleus. Each nucleotide is again made up of 3 components. Very simple, you should remember. DNA made up of nucleotides. Each nucleotide again made up of 3 components. One is deoxyribose sugar or pentose sugar. Second is nitrogenous base. Which are the nitrogenous base? Purine bases such as adenine, guanine. Pyrimidine bases, cytosine, thymine. Purine bases, adenine, guanine. Pyrimidine bases, cytosine, thymine. And the third component is the phosphate group. So each nucleotide is made up of 3 components: pentose sugar or deoxyribose sugar, nitrogen bases, and these nitrogen bases are of 2 types: purine bases and pyrimidine bases. The purine bases, adenine, which is represented by capital A, guanine represented by capital G. Then the pyrimidine bases, cytosine, capital C, thymine, capital T. And the third component is the phosphate group. Structure of nucleotide. As we already know, it is made up of 3 components. We have the pentose sugar, the sugar molecules, the phosphate group, and also the nitrogen bases are there: adenine, guanine, thymine, cytosine. The purine bases are adenine and guanine. The pyrimidine bases are thymine and cytosine. The structure of the nucleotide is showing us that always a purine combines with a pyrimidine. Always a purine combines with the pyrimidine. It gets bonded, connected. The two adenine always combines with thymine with two hydrogen bonds. Hydrogen bond, the weakest bond. And guanine always combines with cytosine with three hydrogen bonds. Very important for multiple choice questions. In a nucleotide, adenine combines with thymine with how many hydrogen bonds? Two hydrogen bonds. Cytosine combines with guanine with how many hydrogen bonds? Three hydrogen bonds. And you should know always adenine gets paired with thymine and guanine gets paired with cytosine. You are observing A for adenine, G for guanine, C for cytosine, T for thymine, S stands for pentose sugar and P stands for phosphate group. This is the structure of nucleotide. DNA, as we already know, it's a macromolecule. It is having the structural and functional units called as nucleotides in many. Hence, it is a polynucleotide. So, the number of nucleotides combined together to form a DNA. This was being standardized and brought to the world with the great efforts of Watson and Crick. Hence, it is been standardized model. Many other DNA models are being available, but scientifically accepted and proved and is being standardized is the contributions of Watson and Crick. So, we call the DNA structure as Watson and Crick model of DNA, and it is a double helix model also. The DNA is a double helix model. You can observe here a twisted ladder. You might have observed ladder in this way. Ladder is there. So, ladder you have seen that. Eni antivi kannadadalli. So, in that ladder you have observed. But this ladder is twisted. It is a twisted ladder. DNA is a twisted ladder according to Watson and Crick. ಸುರುಳಿ ಸುತ್ತಿಕೊಂಡಿರುವ ಏಣಿಯಂತಹ ರಚನೆ ಈ DNA. ವ್ಯಾಟ್ಸನ್ ಮತ್ತು ಕ್ರಿಕ್ ಮಾದರಿಯ DNA, where can observe that the pillars of this is made up of alternative pentose sugar and phosphate, PS, 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 PS. Always it is made up of the pillars of this ladder. Then the steps of the ladder, it is having the nitrogen bases. The steps of the ladder is having the nitrogen bases, and the nitrogen bases adenine with thymine, guanine with cytosine, adenine with thymine, guanine with cytosine, and they are being bonded with adenine with thymine with two hydrogen bonds, guanine with cytosine with 3, adenine with thymine 2, guanine with adenine with thymine, guanine with cytosine, adenine with thymine 2, guanine with cytosine 3. This is a rough model for explanation. You can observe very clearly the model of the DNA here. So, a twisted ladder where the ladder pillars are made up of phosphate and sugar, and the steps of the ladder is having the nitrogen bases. You can observe green color adenine and purple color thymine. Then you are having the light maroon color cytosine, and then we are having the, here this is the purple color for your kind information, guanine, and the phosphate is the backbone. A twisted ladder with pillars made up of alternative phosphate and pentose sugar, the steps made of nitrogen bases, where the adenine combines with thymine with double hydrogen bond, and guanine combines with cytosine with triple hydrogen bond. This is the structure of DNA. Easily you will get 3 out of 3 marks when you start explaining point wise and drawing the diagram also. Dear students, kindly remember, in science, exam oriented, if any question asked for explanation and if it is having a diagram, you have to draw the diagram and you have to start explaining the diagram stepwise. What is the function of this DNA? The main importance of DNA, no doubt it is a blueprint for various life activities to take place. More than that, to our extent, to the chapter related, the role of DNA is hereditary characters are transferred from parents to offsprings in coded form. In coded form, the hereditary characters are transferred. It is the main function of DNA. Genes. Mendel called the characters as triads, and further these triads were given a particular name, and that is nothing but genes. Genes, genes. They are the functional segments of DNA. The genes are the functional segments of DNA. The gene was coined in 1909 by Johansen. Chromosomes are the vehicles of genes. Vehicles or carriers of genes. Chromosomes are made up of DNA and proteins. Genes are the hereditary units which maintain the constancy from generation to generation. So, genes are hereditary characters. They are the functional units of DNA. They are carried by chromosomes from the parents to the offsprings in order to retain the constancy in the chromosome number, and also to transfer the hereditary characters from parents to offsprings. These chromosomes are hence called as vehicles of genes, since they carry the genes from the parents to their offsprings. The genes, if you observe here, the chromosome, dear students, about chromosome to quote, when the cell is still in an immature stage, the chromosomes will be in a chromatin network form. As the cell undergoes maturity and it is ready for cell division, then the chromatin network converts itself into chromosome. Is a chromosome. A single one of this is called as a chromatid. The two chromatids combined, we call this as centromere. Centromere. Sorry. So here I shall, the two chromatids is one chromatid, then the another chromatid. The two combine the place and we call this as the centromere. This is called centromere. So, the chromosome, which is made up of the DNA and proteins, and the DNA is having the functional unit called as genes, and these genes are carried by the chromosomes. Dear students, today's class of the heredity and evolution, the fourth part which we understood was about the DNA and also the genes, where we came to know that the DNA is the nucleic acid that is found in the nucleus, and its abbreviation is deoxyribonucleic acid. And this DNA is made up of structural and functional units called as nucleotides. Number of nucleotides joined together to form DNA, hence it is called as the polynucleotide. Further, we understood each nucleotide structure, where a single nucleotide is again made up of, very happy Sai Krishna and Rahul both are telling the nucleotide is made up of three components. Perfect. One is the pentose sugar. The next is the nitrogen bases, which are of purine and pyrimidine. The third is the phosphate group. Further, we continued with the genes, which are the functional units of DNA. They are the hereditary characters, and these characters were carried with the help of chromosomes from parents to the offsprings. I hope today's class is clear for all of you. Very happy to see all of you seated interestingly, with interest, and also noting down the main points. For better understanding, try to recap in your respective places of the today's part of this chapter and answer the assignment 4 questions with honesty. First question, expand DNA, RNA. Second question, complete the following nucleotide structure and name the parts. You should complete the blanks and also you should name the parts also. Third question, name the following figures and write their functions. So you have to name by seeing these pictures and also write their function. I hope all of you have understood, and surely you all will be ready for the next class of the same chapter, the next part. Till then, have a good time and also pray for the world's peace and happiness. Prayer is the key in the morning and also for the ever success in the life. Faith work wonders, faith in God and prayers for God is itself the grace that will be showered for your effort. Always remember, dear students, effort plus grace is equal to success. Effort also should be there and also God's grace also. Then only the success. Both should go hand in hand. Have a good time. Thanking you one and all. Samasta lokaha sukhino bhavantu. Jai Sai Ram.